So, welcome everyone. Thank you very much to be with us today for this Family Scientists Conference. So just a brief introduction on how to use Zoom. So you can choose your language channel to listen to the presentation in your own language. So you can click on the small globe button in Zoom toolbar where it's written interpretation. During the talks, you can ask questions by using the question and answer tool. The speakers will answer to them during the dedicated time for question at the end, completely end of the presentations. So write the name of the scientist you want the answer. At the end of the session, after the presentation, you can discuss directly with the scientist by raising your hand up, by clicking on the end button in Zoom toolbar. If we have the time, you will be able to speak. When you raise your hand up, you will receive a notification to restore your mic. Note that this is a webinar format, so we can hear you, but we cannot see you. So before starting this first presentation of the day, chaired by Professor Assumpció Bosch, Mr. Guy Alba, Chairman of ELA International Board, will introduce the second day, and Professor Bosch will then take the floor. So Guy.
Hello, everybody. It's a pleasure to be here and to chair this session, which is devoted on genetic and undetermined leukodystrophies, and we will focus on CACH syndrome, megalencephalic leukoencephalopathy with subcortical cysts, Canavan and Alexander diseases. And I'm happy to introduce the 5 scientist women that will present to you their research on this subject. The first presenter is Lucia Lockwitz, who is working at the University of Tübingen in Germany, and she will talk about unveiling new leukodystrophies, the journey to identifying EEF-CEC deficiency.
Professor Bosch, sorry we've missed the presentation of Guy Alba before.
Oh, sorry.
That's fine. Sorry about that. Guy, if you want to take the floor.
Oui, je vais le faire rapidement. Excusez-nous ce petit contretemps.
Very quickly, sorry about this little glitch. So welcome everybody for this traditional meeting held annually between the families and the researchers. We're absolutely delighted to have everybody with us. There will be 4 sessions in total. 2 already have taken place yesterday, and there will be 2 today. And we have different types of leukodystrophies being discussed. However, more than 400 families have registered for this webinar from 35 countries. It's a record, but it shows, it speaks volumes regarding the interest for this meeting, and we're very happy. The meeting will be translated simultaneously in 7 languages. We've added Japanese this year to meet the requests of families in Japan. A friend association called A Future in Tokyo, and they want to attend the webinar. And yesterday, everything really went well, and they were very happy to be able to participate. I'd like to thank the ELA team for having organized this meeting. Audrey Boyer, who is in charge of the research department, Laure Philippe, who's a project manager, working with her, Amé Pierra, whom you don't see on the screen, but who is in charge of the IT-related issues. He's like our firefighter every time we have a problem. Jean-Luc Corti, General Secretary and of the association. And the interpreters team also, I would like to thank the team coordinated by Isabelle Brousset, whom you no longer see on the screen, but she's there behind the curtain with her company called Pour Parler. I thank the interpreters because they're really helping us. and we can break down the language barrier by using the interpreter, so it's very helpful. And finally, I'd like to thank wholeheartedly all the doctors, all the researchers, those who are going to deliver presentations during the weekend, but also those who are working in the field of research and dedicating a lot of time and effort to our patient community. Not only do they deserve to be acknowledged, but the best way we think we can thank them is by funding their work. In 2024, ELA International funded 15 research programs, total 1,700,000 euros. Ever since ELA was set up, we have invested a bit more than 50 million euros in medical research with regard to leukodystrophies, obviously, which means 586 programs we funded. Now, those figures are obviously absolutely essential, but they don't really tell anything regarding how committed the members of our scientific committee are. I want to pay tribute to the work they do, how they monitor the programs, how they allow us to guarantee that the projects funded by ELA International are reliable and high quality partners. And this helps us guarantee the funds also that come from our funding partners. These figures do not pay tribute to the argumentation and thinking work conducted by our ethics committee. In our ethics committee, we have researchers, physicians, ethics specialists, and also parents, families who are directly involved in the association. Recently, the committee has been working on two vital subjects, and by vital, I really mean vital. These topics, or rather, providing an answer, a solution to those issues can save lives. The first topic was the screening, neonatal screening of leukodystrophies. Neonatal screening is the way we call it. And the second topic is access to new therapies. The ethics committee has definitely declared to be in favor of neonatal screening in the whole population for adrenoleukodystrophy and metachromatic leukodystrophy in particular, but leukodystrophies in general. We have written an informed opinion, which was used last year by France to make a request for neonatal screening with the French Ministry of Health. And the request has been accepted. I believe that's because the file was sufficiently substantiated. It will be approved this year by the High Health Authority. It is the first step towards systematic implementation of neonatal screening of all babies born in France. And I really encourage associations, not only ELA, all the associations working with us, to rely on this file and use it. We can transfer it to them if they want it. We can even help them develop their arguments so that they can request neonatal screening with their own ministries of health in their own countries. Now is the right time. Many countries are ready to hear the parents, provided the parents can speak in a loud voice. It is absolutely essential that we work in this direction, and researchers will agree with us. It is important that we screen babies at birth so that we can do something early as possible to treat those babies. Second topic, access to new treatment, new therapies. The council, the board has not yet given an informed opinion, and it's going to be an even more complex subject to deal with. There are very, very, very few new treatments for leukodystrophies, but there are a few. And so when they are available, unfortunately, access is very limited and difficult. And if we want more new therapies to appear, and we absolutely need them desperately, it is important that we support medical research relentlessly. All the more if we consider that medical research is threatened And that's exactly the idea of the Stand Up for Science movement that was recently created in the US, and that is gathering speed elsewhere, including in Europe. We are all opposing the budget cuts that have been introduced by the American government, actually attacking their own research system, which they should be proud of. Medical research in the United States is definitely the most important research program across the world. And I'd like to tell you, physicians, researchers who are working on our diseases and looking after our diseased children and adults, all our hopes lie with you, and you can count on our acknowledgement and gratitude, and we will reinforce every effort this year. You really deserve a round of applause. Thank you very much, and enjoy the seminar, and I hope you're going to enjoy the webinar. And I also wish you a very nice weekend for what is left of the weekend. I will now give the floor to Assumpció Bosch, whom I thank for having.
And now I'm going to share my presentation since I'm the next speaker. Okay, here it is. My name is Assumpció Bosch. I am from the Institute of Neuroscience at the Universitat Autònoma of Barcelona in Spain, and I will talk about megalencephalic leukoencephalopathy with subcortical cysts with a title, Could Gene Therapy Treat MLC? Insights from Mouse Models. I really thank the European Leukodystrophy Association to give me the opportunity to share with you our results. Megalencephalic leukoencephalopathy with subcortical cysts, MLC, was described in 1991 by the group of Marjo van der Naap as a result of mutations in the MLC1 gene. And most of the patients, around 75% of the patients, do have mutations in this gene, and those are like for vanishing white matter recessive mutations. So you need both alleles to bear a mutation to manifest the disease. This protein is expressed in astrocytes, which Marjo also explained the function of these cells in the brain. And these patients show early onset macrocephaly, cerebellar ataxia, spasticity and muscle stiffness, epileptic seizures that, like for vanishing white matter, exacerbate after mild traumatic brain injury or high fever. And sometimes they show mild cognitive decline without autism. But other genes have also been described as causing the disease. This is the case for glial CAM, and these two groups of patients, MLC1 and MLC2A, which are also due to recessive mutations in glial CAM, show a very similar phenotype, but there are also dominant mutations on glial CAM that give also as a result of the disease. Those are called MLC2B. And this means that only a single mutation in one of the alleles would be enough to manifest the disease. Glial CAM is expressed in astrocytes as well, but also in oligodendrocytes and in some neurons. And very recently, two more genes were described as causing the disease, GPRC5B, expressed in both astrocytes and oligodendrocytes, and water channel aquaporin 4 expressing astrocytes. For the moment, there is no clue about the function of these different proteins and their interaction in the cell. So we don't know the mechanism by which the disease is it appears. So there's no a cure nor an effective disease-modifying therapy for MLC patients so far. What we know is that all these proteins are found in the plasma membrane of astrocytes, and they have some kind of interactions between them. For example, if MLC1 is missing in the plasma membrane, glial CAM is not located in this membrane, even if it's produced by the cells. And the same happens with CLC2, which is a chloride channel. So it seems that water ionic hemostasis is very important in one of the altered functions in the patients. So these patients are recognized by or diagnosed by MRI, and you can see here a control or an MLC1 patient. They have a cerebellar hemispheric white matter, which is abnormal. And what is characteristic of the disease is the appearance of these subcortical cysts that are present in the patients, and they usually increase over time. Here we have an MLC1 or MLC2A group of patients that show a progressive increase in this subcortical cyst. Contrary to this group of patients, the MLC2B, remember, those are the patients with glial CAM dominant mutations. They show a remitting phenotype. So they have this abnormal MRI when they are very young, at 1 year of age, and then progressively the cerebral white matter abnormalities decrease over time, and the signal intensity becomes less abnormal. So they have this remitting phenotype. As you can see in this slide, we can clearly differentiate this remitting phenotype, MLC2B patients, from MLC2A or MLC1 patients, where they have a significant motor impairment from their very young age, contrary to the MLC2B. And also the number of seizures is much more increased in the MLC2A and MLC1 patients compared to the MLC2B. So this reversible phenotype in the MLC2B patients suggested that maybe MLC1 or even MLC2A patients could also be reversible, maybe with a small amount of the normal protein, if we can provide it to their brains. So this was the reason why Raúl Estevez, who had been working now in MLC for more than 20 years, tried to convince me 10 years ago that it would be worth to invest our time on gene therapy for MLC. And there are some animal models for MLC. Those are models that do not have glial CAM or MLC in their bodies. They are knockout animals, and they show a mild phenotype of the disease. So histopathologically, we can only see this vacuolating phenotype into the cerebellar white matter, which progresses over time in both models. Here at higher, the ultrastructure can be better observed, and they are considered models for the initial stages of the disease. I must say that the animal models not always manifest the same phenotype as the patient. In fact, usually they do not reproduce the symptoms of the patients, but what we need to look for is in order to use them as models, is try to find which differences can we find between a wild-type animal and a disease animal. So then we can use these features to follow the progression of the disease and the efficiency of the treatments. So these animals can also be studied by MRI, and you can see this vacuolating phenotype is detected in the cerebellum, although very mild. So we decided to use gene therapy to try to get to treat these animals, and gene therapy had been a growing field for many years, and with many different trials, clinical trials that have been approved, most of them in the last 5 years. So it's a really active field for treatment. And I know that you talked about AB vectors yesterday, so you probably know about them. They are quite safe vectors and very efficient. They mostly don't integrate in the genome, so they are safer compared to lentiviral vectors, for example. They do not contain viral genes. This increases their safety, and they are very efficiently transducing non-dividing cells like astrocytes into the brain. And the most important thing is that the treatment can be provided by a single dose, and this achieves long-term expression. There are several treatments already approved in the world, and the most encouraging results have been obtained, for example, for a spinal muscular atrophy with more than 4,000 patients treated over the world. So we first started focusing on the cerebellum, since at that time we didn't know that these animals had locomotor abnormalities. And we use a vector that, just to see the biodistribution, expresses the green fluorescent protein, and we administered it through into the cerebellum using a promoter that allows expression of this protein only in the astrocytes, because we wanted to target only the cells that needed MLC. So then we did the therapeutic study. These animals, they start having this vacuolating phenotype in the cerebellum at 3 months of age. So we did 3 different cohorts. We treated animals at 2 months of age in a preventive study, at 5 months of age. So we call this early therapeutic, at a late therapeutic study when they were already 15 months old. This means 1 year after they develop the vacuolating phenotype. And remember that the mouse only lives around 2 years or 2 years and a half maximum. So we sacrifice the animals 3 months after treatment to look for the histological evaluation. And here you can see that the non-treated animals do not show MLC, and this has the consequence that neither glial cam nor CLC2 are expressed also in the plasma membranes of the cell. This corresponds to the cerebellum, the Bergman glia of the cerebellum. And we can see that these animals have these vacuoles into the white matter of the cerebellum. Contrary, MLC knockout animals that were treated with the AB vector, they show MLC expression, and this was enough to obtain glial cam expression and CLC2 expression in the same cells, very similar to what has been seen in the wild-type animals, and corrected the vacuolating phenotype in the cerebellar white matter. We look at the ultrastructure by electronic microscopy, and you can see that the vacuoles are huge in the oligodendrocytes, and also are present in the astrocytes in the diseased animals, and this is corrected in the treated animals, very similar to the wild types. And we quantified these vacuoles, and we could see that despite the age at which the animals were treated, 2 months, 5 months, or 15 months, 1 year after the appearance of these vacuoles, they are always correcting, suggested that we may have in these patients a larger time window for treatment compared to other neurodegenerative diseases that need to be treated before the symptoms of the disease appear. So as a conclusion of this part, by administering the vectors into the cerebellum of these animals, we could see that MLC is restored in the plasma membrane of the astrocytes, and this also allows the appropriate location of these other two proteins, glial-CAM and CLC2. And we were granted with an orphan drug designation in September 2021 by the European Medical Agency for this treatment. And the other approach, what we decided to do based on this preliminary data I'll show to you, was the transduction of the whole brain, because in fact, the patients, they do not show the most histopathological alterations in the cerebellum, but in the whole brain. And we decided to use a new vector that was described at that time that was able to cross the blood-brain barrier. So we injected the vector into the circulation of these animals, and we achieve transduction in most of, or an important part of the astrocytes in the brain. The cerebellum was not as well transduced, but still we had some expression. So our therapeutic assay was, as described here in this slide. We treated the animals as a late therapeutic study at 10 months of age. At that age, they already show this motor phenotype. In fact, we already see a motor phenotype at 4 months of age in these animals. So we treated them when already had these phenotypes, and we followed them for 1 year, up to 20 months of age. Just before sacrifice, we did MRI studies in these animals, and then we look for the histological evaluation. The idea of this assay was to assay for in vivo indicators of disease correction, to translate it to MLC patients, and also assay for long-term expression of the therapeutic gene. And here is the wire hanger test I showed you at the beginning of the presentation. In dark blue, we have the non-treated animals, in gray, the wild-type animals, and in light blue, the treated animals. And as you can see, for the different parameters that we analyze in these animals, we don't see any difference between the wild types and the treated animals for this test, and neither for the clasping test, which is progressively increasing in the non-treated animals, and we don't have significant differences on the treated animals compared to the wild types. And I must say that they already had this phenotype when they were treated. We also did MRI in these mice. We found that the abnormalities that were found in the cerebellum were also present in the whole brain of these animals. Here, the third column corresponds to the knockout untreated animals, while the two first columns are the wild-type animals, and the last one is the treated animals. So we found this as reddish as the most red is the brain, the more water, the more edema is detected in these brains. So this edema, it was restored after the treatment. And we also did see some abnormalities in the myelin, which is also almost completely restored after the treatment. And this correlated as well with the vacuolating phenotype, that it's also completely restored after the treatment, and very similar to the wild-type animals. And finally, this is the location of MLC1 in the vertebral ganglia of the cerebellum. In the treated animals, you can find it in the last row. The non-treated animals in the third row, you can see that we have MLC expression, and this is enough to appropriately locate glial CAM in these tissues. So as a conclusion, by administering this AB9P31 vector via tail vein, we achieve long-term expression of MLC1 in the brain of these animals, which significantly improve MRI and restored locomotion, which suggests that we may have a wide time window treatment for these patients, and we can achieve a long-term correction. And here is my team and also my collaborators, the group of Raúl Estevez. This work was done by Alejandro, Angela, and Irina in my lab, most with the help of the rest of the team. And the most important thank you to the European Leukodystrophy Association, that for many years it was our only support in this project. And recently we also got funded by the United Leukodystrophy Foundation, and also we have help from the Alliance of MLC. And this is my presentation. I look forward to your questions. Thank you very much, Jeanette, for such a detailed explanation on Canavan's disease. Our last speaker in this session is Elly Hol from the University Medical Center in Utrecht, the Netherlands, and she will talk about an update on Alexander's disease research and implications for patients.
Hey, thank you very much. Can you hear me and see my screen?
Yes.
Yes, that's great. I would like to start by thanking Ela for inviting me to talk about the research that we're doing on Alexander disease. And also thank the audience, because this must be quite a long afternoon, but I hope it is very helpful for many of the patients and patient families. So I'll talk about Alexander disease, which is also a rare disorder, first described in 1949. It's about 1 in 2.7 million. I think this is a number from Japan, but it's a very rare disorder. As you can see again on a MRI, there is these dark spots, and you have seen now some MRIs before, and this means that the white matter is not healthy, also in these patients. The patients suffer from mental retardation, from seizures, from muscle stiffness. And if you look into the brains of these patients, so under the microscope, what you see is these pink dots, these round dots. And if you even go with an electron microscope even deeper, then you see that these are aggregates of a specific protein. What many of the patients also have is a larger brain, so an abnormal large brain, megalencephalic. These aggregates of proteins, they're called Rosenthal fibers, and they are found in astrocytes. And you've also heard about these cells before, because in many other white matter disorders, also these cells are involved. So what are astrocytes? So astrocytes are these green cells in this figure. And these cells are very, very important to support the neurons. And neurons are the nerve cells in blue here that are important to transfer signals, for instance, from your brain to your little toe to move it. And astrocytes will make sure that a lot of nutrients that are present in our blood will be taken up and transported to neurons, so that they have sufficient nutrients to act and to work. And these cells can also be involved in the communication between neurons. So somehow in these cells, there's something happening which leads to these Rosenthal fibers, which we also know are very clearly visible, very close to vessels, blood vessels. So what we know now is that, what we know since 2001, because here you very nicely see a blood vessel, and you see that the astrocytes around that accumulate this abnormal protein. And we know since 2001 that the disease is caused by a mutation in a gene called GFAP. It's called glial fibrillary acidic protein, quite a long name, but it's like a marker gene for every astrocyte in our brain. So what is this protein doing? Because of course, as a scientist and also yeah, as a scientist, you also always want to know how such a mutation or mistake in a gene can lead to the disease. But then you first need to know what this protein is normally doing. And this protein forms like the skeleton of a cell of the astrocyte. So here you see different cells. So that's a very, under the microscopes, it's about 20 micrometers, so very small. And all the green colors looks like a skeleton of a cell. And this is the protein GFAP, which forms very nice, totally fills the cells. And we know now that this protein is important for the structure of the cells, but also, for instance, keep the mitochondria at a specific place in the cells. And those are the little parts in the cell that are important for energy uptake, for instance. And we also know that this part of the cytoskeleton is very much changing when there is a problem ongoing in the brain. So it's a rare disorder, and the mutations have been mapped. And this is one of the figures also out of a paper of Alba Messing, who is a main researcher studying this disease. And you see that the mutations are covering the full length of the protein. There are a few hotspots. So one is here, it's called the arginine at position 239. So there are many, many patients. So every single dot is one patient where they found a mutation. So there are many mutations around this amino acids. There are also a hotspot here at arginine 79, and there are hotspots here in the end. But all these different mutations scattered over the full length of the protein lead to a similar type of Alexander disease. And there are several different types. So very often, I think in 80% of the cases, it starts very early in life. But there are also cases identify where the disease started later or even in the adult life. So what also happens is that if you have a mutation like this, what happens is that normally this protein forms a very nice network in a cell, a skeleton. But once there is a mutation, you get this blobby-like structure, and the protein cannot form this nice skeleton anymore. And you get these accumulations of this protein, which is not in a nice network, but forms little clumps of proteins which should not be there. And we think that these clumps of proteins somehow interfere with the normal function of these cells, thereby leading to problems also into neurons. So to understand the biological mechanism of a disease, and you heard several examples of that this afternoon, researchers always try to build a model. So that is a model that mimics the disease in the patients. And you can do that by making mice with the mutation that we found in patients, or making rats. But you can also do that with models in cells, and do that in vitro, so with a cell in a culture dish. And these models are now gaining a lot of attention because we can now make models from patients. But many people are in the opinion that you need all these different models to gain more knowledge about the disease. So you need the patients themselves, you need also the brain tissue of patients. After death, many people many patients also are willing to donate their brains for research, which is extremely important and can be very helpful to understand what kind of problems are really ongoing in the patients. But also we need a living organism so that we can see, for instance, that if we have a mutation in a specific gene, that these mice also have problems with their motor coordination and other stuff. But we also need these cell models, because that can be the bridge between the patients and the mice models. There's one clinical trial starting in this with this disease, and this clinical trial, it's an antisense oligonucleotide. It's a small piece of DNA that can be injected, and that will interact with this protein that is clumping and somehow will lead to a decrease in the amount of proteins in the brain and therefore less clumps. This has been tested in a rat model of Alexander disease. This is the best model so far because mice do not fully mimic the disorder, and a rat model does that better. And what this group has shown is that this is brain tissue of the rats. This is the wild type rat. This is the rat that has an Alexander mutation. And here again, you see these dark purple dots, which are normal cells, but you see these a bit more pinkish, larger dots. These are these Rosenthal fibers. So this is the abnormal proteins that form clump in Alexander patients' brains. And you see that when these rats were treated with this antisense oligonucleotide, these big purple or pinkish blobs were gone. So that looked very promising. And they also looked at how these rats behaved. So here they got a antisense oligonucleotides at 3 weeks of age, and they looked at their body weight because there was also a big effect on the body weight in these patients. And this line below is the line of the Alexander rat, which did not get any treatment. And these 3 lines on top are the wild type rats, but also the Alexander rat, this one that got treatment. And here you see no difference between a rat with Alexander disease and without. Even after 8 weeks, so if you start the treatment later, you still see that this line here is the Alexander rats that did not get a treatment are much lower in weight in this case than the Alexander rats that got the treatment. So you can also start the treatment later in life, which is also promising. This is one other test because in this paper they have many other tests, but also what you see here, that's the grip strength. So they check how strong the rats are holding on to a wire, for instance. And here you see that so the pink rectangles that are the Alexander rats, and you see they are much less. They are not so strong compared to the controls, and this is the control treatment. But if they get the antisense oligonucleotides, it normalizes a bit. So this looks quite promising. There is an Alexander trial started, and it's also, I know that also Professor van der Knaap is part of it, and that they're also including patients in Amsterdam. And there's no outcome yet, but I'm very curious about the outcome for this trial. So going back to these models, so the rodents are very important in the studies, but they are not men, and there could be essential effects that we could not find in rodents, but that we can find in cell models. And over the years now, you see a huge increase in the number of cell models, and that is based on a breakthrough in science where in 2006, this person, his name is Shinya Yamanaka, was able to make stem cells from skin. So from every person, also from me, but for every person in the world, you can simply take a little bit of skin or a little bit of blood, and with a certain technology in the lab, you can make stem cells of that. And if you have stem cells, you can create any type of cell in the body. You can create liver cells, muscle cells, but also brain cells. And these stem cells are called induced pluripotent stem cells. It's quite a long name, but it's being used now quite a lot in the lab. And this was such a breakthrough that 6 years after the first publication, he got the Nobel Prize for Physiology after 6 years. So it's really something that has revolutionized science. And I'll show you some examples for that also for Alexander's disease. So when you have these stem cells that you can create from a bit of blood, for instance, what you can do in a Petri dish is make different types of tissue and also brain cells. And in 2013, so about 7 years after the first breakthrough that we can make stem cells from human skin tissue, there was another breakthrough, and that is that Madeleine Lancaster and Jürgen Knoblich from Austria, they showed that if you start with these stem cells and if you put them in culture, then you can grow them in mini brains. So you can make mini guts and mini liver, for instance, but you can also make mini brains. So it's not a real brain, but it's clumps of cells that start looking like brains, and they have the cell types that normally you have in a brain. So these type of mini brains you can use for looking at developmental disorders in the human brain. And how you make them is that you can make them from blood or from skin, and then you can reprogram them. So that was why they got the Nobel Prize, because they found out how you could reprogram these skin cells into stem cells, and then you grow them for several days by adding all kind of different growth factors. And in the end, so first you start with a layer of cells, but they grow like so-called embryoid bodies. And in the end, you get these little balls of cells in a culture dish, which can grow as big as a green pea. And if you look inside, you find neurons and astrocytes and oligodendrocytes. So these are these cells. So this is a petri dish of 6 centimeters in diameter, and these are quite old organoids, brain organoids growing here. And in the lab, these organoids have to be in constant move, because we need to try to get all nutrients to the center of these cells, and that's not always very easy. But over the years, we have better and better technologies to grow these cells even better and better. And this is, you can stain them with all kind of different antibodies, and then you can look them under a microscope in 3D, and then you can start asking questions or try to learn something new, which you probably did not see in the rodents, but you can see some of these things also in these clumps of cells. But of course, you can never ask whether this clump of cell can hold better to a wire, because it's a clump of cells. So the functional testing is a bit more is more difficult or impossible in these clumps of cells. So these brain organoids now are made for several diseases, but in my lab we got stem cells, or iPSCs they called, from several patients that had Alexander disease, and we repaired the mutation. You can do that with CRISPR-Cas. That's another recent technology. So you can repair the mutation, and then you have a control that has exactly the same genetic background, because it's from the same patient, but the disease mutation has been repaired. We did that with several different mutations. And then we started, what we wanted to see is whether the development would be different. And we expected, of course, to find all these Rosenthal fibers in our mini brain in the dish. But that's not what we directly found. What we did find was that our controls, so here are 3 dishes with control organoids or mini brains, and on top these are of Alexander patients. So one of the things we did find back was that Alexander organoids grew bigger. So that is a bit similar to the megalencephaly that the patients have. And we saw under the microscope, so this is when you make very thin slices of these organoids, we saw that they look differently. So these are Alexander organoids, and these are controls. And here you see a larger hole, and this is an area where new neurons are born, and this is much broader in a control situation. So something was happening with development in these early organoids. And this is not a very beautiful picture, but what it does show, again, on the top is the control, and here below is an Alexander patient organoid. You see lower number of pink cells, and these pink cells indicate that in these organoids we have a lot of neurons and a lot of neural tissue. So somehow something goes wrong, and with the differentiation of these stem cells into real brain tissue in this case. And that's also what we found, and this is one of the last slides that I will show. But if you have these organoids, so here again, after 165 days in culture, so we can culture them for a long time, even more than a year. And you see that the Alexander mini brain is really larger than a control mini brain. What you can do then, there's now a technique that you can look in these organoids at the full array of RNA that they transcribe in every single cell. And what you see here is that if you do that, you get like different groups of cells that are in the brain. So these green ones are all the neurons, and these pink ones are the astrocytes. We have here microglia and other cell types. And if we did that with the Alexander patients, the neurons were there, but in the middle they were more immature, and the astrocyte cloud, which is here, was not present. So we found a lot of immature astrocytes. And the very weird thing that we saw, but this might be an artifact, so something that only happens in a cell culture dish, because we have never seen it directly in patients. But in the cell culture dish, what we found is that these mini brains had a lot of muscle cells and a lot of pancreas cells. So what this told us is that if you have a mutation in GFAP, that in this system, you see that there is a problem with differentiation of the cells. So in conclusion, so what we find with these brain organoids is that they grow larger, so that mimics this megalencephaly. We did not find clear Rosenthal fibers, but it might also be that we have to wait for longer. But in the rodent model, this was really clear. But we found a lineage commitment problem. So there was a delayed differentiation, but we also found very weird cells that should not be in the brain. So let me see what so these human organoids are very interesting to study. You could use them for personalized medicine, because you can just take these cells from different patients, and every patient has a different mutation, and then check whether a certain medication will work for all these patients. But they model the disease in a different way. So we cannot answer all the questions that we have only with these human models. We always need to do that combined with studying the patients or studying these different mouse and rat models. But the human models can be very instrumental at the moment that we have a drug that will work, that does work with a certain mutation. It's much more easier to screen, let's say, 20 or 30 different mutations in cells than to make 20 or 30 different mouse lines with these mutations. But the rodent models can be used for more in-depth screens and efficacy of the drugs. So then I think I will be at my last slide. So this research was done in a European consortium with people from Sweden, from the Netherlands, from the Czech Republic, from Spain, and from Israel. And also ELA was a part of this consortium, and also VKS is a Dutch organization. Besides that, we have a separate grant on La Caixa together with Dolores Pérez Sala, the Spanish partner, to continue our research. And this research was funded by the European Joint Programme on Rare Diseases. And at this moment, I'm together with Marie-Ève Tremblay from Canada. We are also funded by ELA to have a 2-year project where we're going to study these organoids in even more details at the electron microscope level, and these are a few examples. And we are going to look for effects on myelination and see whether we can find early changes there. And we already found some changes, such as here in the mitochondria, which are the small organelles in a cell important for energy. And with this last slide, I would like to thank you for your attention, and I'm also available for questions, as all of us are after this session. Thank you.
Thank you very much, Elly, for such a didactic and complete update on Alexander's disease and the technology associated with the research of this and many other diseases.
Oh, okay. Did you hear me?
Yes.
Okay, cool. Thank you. Françoise Piguet, please.
So what would you prefer? I should speak English or French?
Yes, please, in English.
Okay, perfect. So thanks to Ella to have invited me to be presenting in this session, but also to be the chairwoman. So let's do like this. I hope you will see my screen. Could you just confirm that you can see well my screen and it's full screen?
Yes, we see it.
Perfect.
Thank you.
So the idea today is to give you an overview of let's say, in the program was written gene therapy for metachromatic leukodystrophy. However, as we are also developing other things in the lab, I want to more speak about new therapeutic development for metachromatic leukodystrophy. So first of all, for some, we just saw some talk about gene therapy, but I want to be sure that everyone can understand. And so what is the principle of gene therapy? The objective is to be able to introduce a therapeutic gene in all the diseased cells of the body. But how can we efficiently target the diseased cells? And for now, the best thing we have that could be a Trojan to go to the cells are viruses. However, how is a virus working? Because you all well know, I think, virus since now we faced a COVID pandemic. And so a normal virus, what we call a wild-type virus, will go to the cells, multiplicate into the cells, as you can see here, and finally kill the cells to be released. And this is not absolutely not a virus that we can use as a therapeutic purposes. So what scientists have done is that they changed part of the virus. We kept what we call the capsid, which is surrounding the virus, but we changed the gene which is inside by replacing the virus's gene and putting a therapeutic gene, so that the virus will keep its capacity of a Trojan to well infect the cells, will deliver a therapeutic gene, and so we can pass from a diseased cells to a corrected cells. Maybe I should put my pointer, which would be better. So what kind of gene therapy can we do? I think all of you are familiar with what we call ex vivo gene therapy, which are the base of several therapies that have been done for adrenal leukodystrophy, but also more recently for MLD with the LibMLD therapy, so that you can take the CD34 of a patient that you will go to the, put the CD34 in culture, infect with a lentiviral vector, and reinfuse the cells to the patient, and part of these cells will migrate into the brain to be progenitor of microglia and will produce the desired gene, and can be done for MLD, for MPS, for Krabbe, and for several other leukodystrophy. The other solution is to do what we call in vivo gene therapy. And in that case, we will directly inject the virus in vivo, either using intraparenchymal delivery that has been done in the past by our team for MLD, intravenous gene therapy, as some of the patients were speaking before with AV9, and it's already approved in clinic for SMA, or intrathecal delivery. So coming back to metachromatic leukodystrophy, I think that most of you are familiar with. It's a deficit in aryl sulfatase A. The ARSA is an enzyme that catabolizes the sulfatide to galactosylceramide. And what occurs in patients lacking for ARSA is an accumulation of the sulfatide in all cells of the brain, like the neurons, the oligodendrocytes, the macrophages. And consequently to this accumulation, the cells will degenerate and die. An advantage of the lysosomal enzyme as the ARSA is its capacity to be secreted and uptake. As you can see here, part of cells that are able to produce ARSA would secrete ARSA outside of the cells. This
enzyme could migrate everywhere in the brain or even in the periphery and can be uptaken by distant cells thanks to the mannose-cis-phosphate pathway and be then directed to the lysosome where it can be active. And you will see that this is really important for one therapy that I will speak after. So in the lab, and thanks to the team of Volkmar Giesenmann in Germany, there have been an MLD mouse model that we call HARSA knockout that has been generated that have no HARSA expression and accumulate sulfatide as it is in the patient. You can see here, this is a normal mouse. You don't see any blue staining. Whereas here in an HARSA knockout mice, you can see that you have a huge accumulation, especially in all parts of the brain in which you have white matter. What we also demonstrated, even if it's late on, we have a significant behavior impairment in the mice if you compare here the wild type and here the HARSA knockout. And what is occurring after sulfatide is that you will have a neuronal loss, as we can see here with Purkinje cells. You see here normal Purkinje cells and loss of the cells in the mouse, microglial activation, as well as astrogalloidosis that are two markers of neuroinflammation. And we will also come back to that. So the first thing is you all know, and I will not come back on that we started with Professor Aubourg on Caroline Sevin years ago, was during my PhD, an intracerebral delivery of gene therapy. We go to clinic, we had 4 patients included. We demonstrate that was safe, but we didn't succeed to prove a significant improvement in terms of therapy. And the question was, okay, how can we improve our AAV to have a better transduction in the central nervous system? And when I see central nervous system, you have to keep in mind that it's both brain, spinal cord, and then even to the PNS, the ganglia, and the peripheral nerves. So for that, we tested a new vector, which is called AAV-PHP-EB. You can see here images, and I hope the video are good, which correspond to what we obtain when we are doing an IV delivery of such vector. And coding here was for fluorescent protein, and we transporized the tissue, the brain, and the spinal cord to see all the cells that were positives. And also, because usually in presentation, you're more used to see this kind of staining, we demonstrated that we have a huge targeting of the motor neurons in the spinal cord, but also in the brains, as you can see here, with all the small brown dots that correspond to cells that are transduced. And in fact, when we score, we are able to target with only one single IV injection up to 98% of the neurons of the brain. So we started now 4 years ago with an initial proof of treatment in which we took the mice at 6 months of age, in which they are already carrying the disease. So they have sulfatide accumulation and neuroinflammation. And we inject them at 6 months and we keep them alive for 3 months. We collected the brain and the spinal cord. And the first thing we looked at was the sulfatide. You see here, normal mice, cortex, corpus callosum, which is a white matter part of the brain, and spinal cord. You can see here in the HARSA knockout non-treated, so you see this accumulation that I showed you before. And this is what we observe in the knockout mice that received the AAV. And by scoring all over the brain and the spinal cord, you can see that we had a tremendous decrease and almost a full correction of the sulfatide storage in all the treated mice compared to the non-treated animals. We also demonstrated that we have a significant effect on both the astrocyte, as you can see here, in brain and spinal cord, but also the microglia. And at that time, we questioned whether can we still be efficient if we are treated the mice later on. And so when they are more affected, could we still have an improvement? But we also questioned whether if we treat at the same age, could we lower the dose of injected virus to still have an effect? And what we here also demonstrated is that, once again, you can see the staining that we obtain in the brain. So all the red things are positive for the virus. So it's just here the virus. So you can have even more protein going secreted. And here is the spinal cord with a zoom over here. This is a zoom of this part of the brain, this of here, and here, here. We also, because it's an important thing, you heard before speaking about the SMA trial with an AAV9. One of the main problems with the AAV9 by injecting intravenously is the huge targeting of the periphery, and especially the liver, where it can be toxic. In our case, what we demonstrated was that, so here, all these are part of the brain. This is the spinal cord. So these are the different group I told you, the early treatment, low dose, high dose, or late treatment. You can see that as an overall in the brain, we have around 3 copies everywhere in the cells, and the same in the spinal cord. But if we look at the liver, we have less, we have around 0.03 copy maximum, and even less in all the peripheral organ, which is good because it means that we don't risk to have any peripheral toxicity. Sorry. And what we also succeed to demonstrated was a significant increase of the ORSA activity in all the group treated with the AAV compared to the wild type and untreated animal in black and gray, and in all parts of the brain and the spinal cord. Then for sure, we look at the efficacy on sulfatide. So you can see again here, wild type animal, several parts of the brain, spinal cord, knockout animal with the accumulation. And here you can see early treatment, low dose, early treatment, high dose, late treatment, high dose. And what you can see is that in all this group, we completely correct the sulfatide accumulation. And we wanted also to go further by looking at the different isoform of the sulfatide using MSMS, meaning that we can quantify the sulfatide based on the length of their chain. And what we know is that the C18 are mostly present in neurons and astrocytes, whereas long chain are massively present in oligodendrocytes. And what you can see, so here, wild type, non-treated, treated. Wild type, non-treated, treated. We can rescue also by quantification all the accumulation of sulfatide. And according to that, we also look at the neuroinflammatory marker in the brain and in the spinal cord. And we also here demonstrated that in all the group we evaluate, we had a full rescue of the astrogliosis and microgliosis. But now you can tell me, okay, this is in mice. Would this vector still be efficient in large animal model? And so for that, we ran an experiment in non-human primates. And this is an example of what we get as staining. So all the brown staining correspond to positive cells within the brain. So you can see here, caudate, cerebellum, corpus callosum, but also in the spinal cord, in the DRG, as well as in the sciatic nerve. So we can efficiently deliver high amount of ORSA everywhere in the body. We were also able to quantify the ORSA activity. And so for that, we look at the cerebrospinal fluid. And you can see that at baseline, we have no activity in the monkey, either the control or the treated animal before treatment. But after 3 weeks and even more after 6 weeks, we have a significant increase of the ORSA activity in the CSF. We also look at what was going on in the tissue. So when here you have no pink color, is just because this region of the brain have not been quantified. It's not that they are negative. You can see the dotted line, which correspond to the basal levels we have, because you always need to keep in mind that our non-human primates are not affected by MLD. So they have basal ORSA activity. So what we want to quantify is an overactivity compared to the baseline. And indeed, what you can see with all these bars going up to the basal level is that we efficiently demonstrated an increase of the ORSA activity in all the tissue, except in the peripheral nerves. But can we still improve our AV vector to transduce CNS? So this were really encouraging, and we are now moving on to try to build a clinical trial. But just to show you what we are also doing in the lab, sorry, here should have been a V, we can play with ultrasounds, and in fact with what we call focus ultrasounds. So what you need to have in mind is that in your brain, you have what we call a blood-brain barrier. That is protecting your brain from invaders that can be either the immune cells, the virus, and all that. And these vectors, let's say this blood-brain barrier is surrounding the brain capillaries, and you need to imagine that it's something really tight that protect to accept the nutrient and things that are circulating out here inside in the blood to go to the brain. What we can do is to inject microbubbles in the circulation. So you imagine that here you will have your blood vessels. You have microbubbles coming, and when you apply ultrasounds, the microbubbles will start to oscillate into the blood vessels. And what will occur is that with that oscillation, they can transiently open the blood-brain barrier so that the virus can more easily pass the blood-brain barrier and transduce the brain tissue. And this is an example that we've done here is a project more focused on Parkinson, but we can then use the same for metachromatic leukodystrophy. You can see here in red an hemisphere in which we did not apply any focused ultrasound. On green, this is where we applied focused ultrasound and we inject gadolinium. So all these hyperintense signal that you can see correspond to entrance of gadolinium coming from the blood to the brain, demonstrating that we have an open of the blood-brain barrier. And indeed, when we look a few weeks after in the brain of the non-human primates, and if we compare without ultrasound and with ultrasound, and this is a zoom of that part over here, you can clearly see that here we have, and here we have enrichment of cells due to better transduction of the virus because more virus pass the BBB, transduce the cells, and can express the cells. So this is something we are working on right now, and we should see, and we would like to see in the next step, how we can improve for MLD such kind of approach. To finish, I would like to thank all the people involved in this study. Of course, all my team at the Genove, at the Paris Brain Institute, but also our collaborator working on the pathophysiology with Marianne Koll, the team of Ulrich Massner and Volkmar for the mouse, our partners that produce the AAV vector, and our partners for the blood-brain barrier opening, and especially Jean-François Aubry, as well as the platform in the Institute, but of course, our sponsor with Ella, Jensen, and the ANR, because otherwise we cannot run such experiment. And of course, I thank you for your attention, and do not hesitate to ask questions after. And so now I will just introduce the next speaker. Sorry, who is, I will not say any mistakes. It's Yanick from Scotland. And I think if I well understood that Yanick will speak about the Aicardi-Goutières syndrome. Not sure he has a presentation, but he wants to speak on what they are doing right now on this syndrome. Yanick, the floor is yours.
Okay, thank you, Françoise. So I have 20 minutes, I think. Is that right? Oh,
yes, true. You have 20
great.
minutes.
Thank you. Okay, so thanks very much. And hello to people I know and people I don't know. I see a couple of names that I recognize on the call. I was going to keep this very informal. I don't have any slides, actually, which is a slightly peculiar way to do these things. But and I was rather hoping people might be able to just ask me questions and I could maybe conduct the session that way, but I'm not sure if that's going to be possible. So I'm a clinical geneticist. I've been working on Aicardi-Goutières syndrome since 1999, and my group, and in collaboration with other people, have identified the 9 genotypes, the 9 different genetic forms of Aicardi-Goutières syndrome that we can see. And since that time, we've also been trying to define the natural history of the disease and to think about treatments. So in terms of, I'm working between Edinburgh and the Institut Imagine in Paris, but I have in Paris now, my work is more and more being taken over by or taken on responsibility for that work is being undertaken by Professor Marie-Louise Fremont. And I'll just sort of mention that Marie-Louise runs a national French multidisciplinary online virtual clinic once a month, where essentially all cases of Aicardi-Goutières syndrome in France can be discussed. So for at least for French families, that's something that may be relevant. So I think most of you on the line will know that we think that Aicardi-Goutières syndrome is primarily caused by an upregulation of a protein or a set of proteins called type 1 interferons. And type 1 interferons are they're antiviral. So when we are infected by a virus, our cells want to fight the virus, to kill the virus, and to do so they secrete these chemicals called type 1 interferons. And these type 1 interferons are extremely powerful, and they're pretty effective at killing viruses. But the problem is that they're also quite toxic. And in Aicardi-Goutières syndrome, the quite peculiar and slightly remarkable thing is that the body, the cells of a person with Aicardi-Goutières syndrome confuse their own nucleic acid, their own DNA, and their own RNA for that of a virus. And so they produce chronically all the time, they produce this type 1 interferon. And like a disinfectant, disinfectants are good, but only in the right place at the right time for a short period usually. Whereas here, we're producing this biological disinfectant, this stuff that can kill viruses, but we're producing it all of the time, or people with Aicardi-Goutières syndrome are producing it all of the time, and that kills cells and is damaging, and most particularly, and those of you who've got children or aware of Aicardi-Goutières syndrome, that most particularly affects the brain and the skin, although there are other features of the disease. So, for example, people can have mild derangement of their liver enzymes, as just one example. So, because of those insights, we have been interested, we and others have been interested in the idea that you might either reduce the amount of interferon that's produced, or you might block its effect. So you'd still be producing the same amount, but you would in some way block its actions. And interferon is these interferons are very powerful chemicals, and they cause a lot of changes in the body. They're like little generals. They can command a lot of different aspects of the body to do things, and it's those things that are done in response to the command for the interferons that cause the damage. Now, what we know about type 1 interferons is that they signal to have an effect on other cells, they have to attach to a receptor. And there are chemicals that can effectively block the receptor. And some of you will be aware of these so-called JAK inhibitors. And the JAK, the JAK, there's a particular JAK called JAK1, and JAK1 forms part of the interferon, type 1 interferon receptor. And so there are these JAK inhibitors that inhibit the receptor. And people have been using those in Aicardi-Goutières syndrome now for probably for maybe 6-7 years now, or maybe a little bit longer actually. And I would say what we've found is that they do work. They work particularly. It's most obvious that they work at the level of the skin, and you can see skin. It's visible, And using these drugs, we can clearly see changes in a lot of people who have skin problems who are treated in this way. We see changes that are sometimes quite impressive. So my interpretation from those observations is that these drugs do have an effect, and that they give some evidence in favor, if you will, of the this hypothesis about the biology, about the causes of AGS being related to type 1 interferons. However, I think what, for me at least, has been, well, and I think in my opinion, the drawback with these drugs is that they have not been so effective at treating what really is the primary aspect of AGS that we would like to treat, which is the brain disease. And I think that there are a number of potential reasons for those those shortcomings, the fact that it doesn't work quite as well as we would hope. And I think one is actually that by the time the diagnosis of Aicardi-Goutières syndrome is made, usually, and by the time people have the results of genetic testing, then many weeks, if not months, have passed. And so by the time you start the drugs, quite a lot of damage to the brain has already happened, so that we're starting at a time slower than we would like. We'd like to start really with these drugs as soon as possible after the first symptoms, or ideally even before those symptoms arise. And that's a challenge. That's a challenge to make the diagnosis clinically by the doctors, and then to get a genetic diagnosis. The other major reason I think why there might be a problem with these drugs is that they don't penetrate into the brain as well as we would like. So when we compare the amount of drug, if we give a person one of these drugs, there are different types of these JAK inhibitors, but when we use these drugs and we measure the amount of drug in the blood and then compare it to the amount of drug in the cerebrospinal fluid, which is that liquid that bathes that surrounds the brain and the spinal cord, then we find that the levels in the CSF, or the spinal fluid, are only about 10% of the levels that we observe or record in the blood. And I think that is a problem. And so what I think we need to be aiming for in the future is to have earlier diagnosis. I think that is important, and I don't think that's insurmountable, because genetic testing will become quicker, easier. But actually at the moment it's largely a bureaucratic issue. The technology is there, the ability to generate genetic information and diagnosis is trivial now, but doing it at scale across a country in the context of a health service is more challenging. And then the second thing is we would like drugs that penetrate into the brain better. So it's unfortunate, and I don't know how many people will be aware, but there was a company, Biogen, who were interested in developing a drug that would be injected into the spinal fluid, so it would have more direct access to the brain. But unfortunately, that was another blocker of this interferon receptor. But unfortunately, that I actually don't know why, but it was decided by the drug company that they would pull that, or they would stop that program. So that's been something of a sadness, and I don't know what will happen with that work, because quite a lot work was done up until that time. I'm hoping and hopeful that other companies will consider that kind of approach, where we can get closer to the brain by injecting the drug directly or into the spinal fluid, or having drugs that cross more easily into the brain. The brain is highly protected from an immunological perspective. It's a very delicate organ, so that the body has these sort of defenses, and one of the kind of drawbacks, or one of the, in inverted commas, side effects, if you will, of these of those defenses to protect the brain, is that actually they stop a lot of drugs getting into the brain as easily as we might like when we want to treat a disease of the brain. The other thing I will just mention is that we previously, with funding from Ella, had published a paper and did some work with so-called reverse transcriptase inhibitors. And we generated data that suggested that these reverse transcriptase inhibitors, which have been used in HIV or for AIDS, in fact, we were what's called repurposing those drugs for the treatment of Aicardi-Goutières syndrome, and they seem to have an effect on the levels of interferon. And very recently, we reported on a clinical trial using the same kind of drugs, but this time the trial was performed in the UK. And very frustratingly, there were so many bureaucratic hurdles to that trial that we effectively generated no meaningful data. And so, unfortunately, I'm no further forward in knowing for sure whether those drugs are effective or not. What I can say is that although we saw levels of interferon alpha fall in the first trial we did, I never really heard from parents that they felt that the drugs really made a dramatic difference, and I never saw any obvious changes, for example, in skin in the same way that we've seen with the JAK inhibitors. So I think at the moment the only drugs that make sense and have some proof of a favourable effect in AGS at the moment are these JAK inhibitors. There is another drug that's coming into use and will be being used I think is starting to be used in some patients with AGS, which is another drug called anifrolumab, which actually blocks the interferon receptor much more effectively than the JAK inhibitors, and really completely limits the amount of interferon signaling in the blood, and so it's a better drug, I think, than the JAK inhibitors. And I think people will be using that in AGS. The problem again with that drug is it doesn't cross very well into the brain, and so I think we're going to still be left with the same or some aspect of the same problem there. Otherwise, I think some of this news will be disappointing for some of the families, and I'm sorry for that. I think we have moved a long way since I first started working on the disease, and having tests available is important and helpful. But the leap to effective treatments has been slower than one would have liked. However, I think that we have learned more about the interferon signaling. We do have drugs that are effective in some aspects of the disease, and I think that also gives us information about the biology of the disease. And what I would also add is that there are lots of drug companies that are very interested in the biology of Aicardi-Goutières syndrome because of this whole issue around self DNA and RNA triggering of an antiviral response, and so there is a lot of research, and I'm hopeful that in the next 3-4 years there will be other drugs on the market that can be considered for use in AGS. I think I'll stop there. I'm happy to take questions if there are any, or maybe that's not possible. Thank you.
Thanks a lot, Yanick. I think anyway, we will have all the questions at the end. So next presenter is Alessio Cantore from Milan, I think, who will speak about how to do an in vivo gene therapy for Krabbe disease. Alessio, the floor is yours.
Thank you. Hello, everyone. Good afternoon. Let me try to share my screen. Let me see if you see correctly. I'll try to minimize this. Okay. So I hope you hear me well and you can see my screen properly.
Yes, Alessio, we can.
Okay, perfect. Thank you very much for confirming. Okay, so we'll go through a little bit of some of our work trying to develop in vivo gene therapy for Krabbe disease or globoid cell leukodystrophy. So this has been already introduced by Dr. Piguet before. So gene therapy is in vivo gene therapy can genetically modify the genetic information of the liver, for example, of the liver hepatocytes, which are the main cells in the liver, through vehicles or derived from viruses, what we call the viral vectors. This has been already introduced before. So these are shuttles that can deliver a functional copy of a gene into human cells potentially. And this has been already achieved by AAV vectors, indeed, that can be administered intravenously and then can genetically modify hepatocytes, the main liver cell type, into our body, and copy and introduce a functional copy of a gene that is missing in a genetic disease. Such as, for example, hemophilia, which is a deficiency of clotting factor, which are produced by the liver and secreted into the blood. And these are already commercial products, so a gene therapy that is commercially available. And AAV vectors work very well in quiescent cells, but if the cells duplicate, if the cells proliferate, such as during growth, it's likely that these vectors are progressively lost because they remain associated to the nucleus but not integrated into the genome. And since actually most genetic disease of the liver have their onset in childhood, we think that ideally we should modify the liver in the patients as soon as possible and ensure maintenance of the genetic modification over time. So this can be achieved, for example, by using different vectors than AAV. One is lentiviral vectors. We already heard about. Dr. Piguet also alluded to the use of lentiviral vectors for transducing hematopoietic stem cells, cells making the blood. So we can use the same vectors essentially in vivo and potentially genetically modify the hepatocytes and maintain these, because the vectors integrate into the genome, maintain the genetic modification in the liver during growth. So that's mainly the work of our lab. Coming to Krabbe disease, as all of you know, this is a neurodegenerative lysosomal storage disorder due to the defect in a single gene, which is a galactosyl ceramidase gene, which is involved into the degradation, the metabolism of some components of the myelin, of course, the lipid shell that is around our neurons. And unfortunately, this leads to progressive neurodegeneration, and the severe form are very severe and rapidly, unfortunately, progress. And thus there is an unmet medical need in this space. And of course, therapies have been attempted, including gene therapies that have been developed in animal models with promising data, and a clinical trial is also ongoing, including AAV intravenous delivery to target the systemic disease, let's say, coupled with cord blood transplantation, which might help the other features of the disease such as in the central nervous system. However, there's still the big challenge is, of course, to deliver enough GALC activity into the brain and the peripheral nervous system. So actually, in our group, we work on the technology to genetically engineer, to genetically modify the hepatocytes stably. So we have worked with lentiviral vectors, as I anticipated, and we have engineered the vectors to really escape innate immunity, so that they really efficiently and safely, hopefully, deliver the gene into the hepatocyte, the main liver cells, and then they can express the transgene. We have already demonstrated stable transgene delivery into both newborn and adult mice, and also in large animal models that are promising, and we are planning actually a phase 1-2 trial in an inherited metabolic disease as soon as possible. So the idea here is to exploit the liver as a factory to produce potentially GALC into the blood and potentially spread it all over the body. And so actually convert the liver into a factory of GALC. Of course, this comes with a challenge, as this already been mentioned previously. Of course, even if we are successful and we engineer the liver to express GALC, unfortunately, it's unlikely that this enzyme will go to the brain and also to the peripheral nervous system. So what we are doing and other groups are doing is try to engineer these enzymes to cross the blood-brain barrier, which we already heard about. It's a barrier made by different cells which really protects our brain, but also it's a block for drugs that we want to reach the brain. So, and there are ways these barriers is crossed. So we are trying to engineer the transgene to allow potentially or to increase the efficiency or to improve the BBB crossing. And also we can play with some signals that improve secretion and production from the liver into the blood. And we hope that maybe one day we can have an enzyme that can cross the BBB. And so this is potentially with a once and done therapy, potentially in the future to use the liver to express these enzymes that can spread throughout the body. And this can actually be also helped by a mechanism that we already alluded to, that is a cross correction. Lysosomal enzymes have this signal that allows some cells to internalize the enzyme. These are some of the data that we have produced. We have engineered some forms of GALC. Here is an example. We are delivering through these vectors into cells in culture, in vitro, the hepatocyte-like cells. We can engineer the GALC to, as you can see, we can effectively engineer GALC to be highly produced and also highly secreted from the cells. And then we can also, so from this data, it seems that this engineered version is the most promising. We can take these supernatants, so what these cells made, and put into neural cells from the GLD mouse and actually demonstrate that we can cross-correct the cells. So from the medium of the cells that produce enzyme, we can cross-correct cells that are defective for the enzyme. And indeed, we can find activity into these cells that wouldn't have otherwise. And again, confirming that this engineered version of the transgene it seems powerful in correcting these disease cells. So this is all in vitro. Another thing that we can try to do in vitro is try to mimic, to model the blood-brain barrier. We can use a device which has which shields, let's say, layer of cells, and we can put endothelial cells on this layer. Of course, this is a simplified model of a blood-brain barrier. We're still missing cell types. We are working to improve models or through collaboration from other researchers that have BBB models in vitro. But again, also through a mechanism of cross-correction across these cells, we show that we can actually cross-correct these cells even through this simplified model of BBB. Here we didn't observe an advantage of the engineered GAL-C actually, but we can show that anyway we can cross-correct these cells even across this membrane, let's say. So going to in vivo, there is a mouse model of GLD, of Krabbe disease. The model is very severe, is a genetically knockout, so it's genetic inactivation of the GAL-C. The mice is very severe, again, only lives for 40 days after birth. Gene therapy, in vivo gene therapy with lentiviral vectors can prolong a little bit the survival. This is not, of course, you know, substantial prolongation, but it's still promising. The model is very, very severe. It's actually, this is another limitation that we probably have. We are trying to work with models that develop the disease a little bit less rapidly, so they would allow potentially to mitigate the disease progression. Anyway, what we have shown is that we can, by intravenous administration of lentiviral vector into newborn mice, we show that actually this engineered GAL-C transgene is actually showing very, very high activity in the liver and also in the blood. This is a serum from the blood. This would be a normal mouse. So you can see we are really expressing a lot more than a normal mice. So we are really making this liver pumping out GAL-C into the blood and the serum. So this is promising, but once again, unfortunately, activity in the brain is not completely satisfactory because it's not fully normal. Still, it's about a fraction of normal. It's about one third of normal. So it's not absent. It's there. There might be some advantage of the engineered transgene, even though this needs to be confirmed and further improved. And also we see some activity in the peripheral nerve, also not fully satisfactory, I must admit, but it's promising because we are really making these mice producing a lot of enzyme, and of course this is not completely satisfactory into the brain and nerves. Anyway, underlying once again the challenge of widespread GAL-C activity into the central and peripheral nervous system. So there are, of course, the work is still ongoing. We can improve also the vector to better transduce hepatocyte, even if the gene transfer, I would say, is very efficient. We can still work on the vector to make them better. We can work on the transgene we keep trying a mechanism of crossing the BBB. So this comes, I would say, this brings me to the conclusion. So we have successfully engineered the GAL-C transgene to enhance secretion and cross-correction. We show evidence of transfer of GAL-C activity from cultured cells into cells that are defective. We demonstrate prolonged survival and supranormal Galsi activity in a severe mouse model of the disease. However, we still have suboptimal therapeutic activity in the central and peripheral nervous system, as I've shown you. So further improvements are needed, but I think the work is promising. We definitely need to improve the transgene to cross the blood-brain barrier. The blood-brain barrier, it's really a big issue for delivery into the brain. It's what protects our brain, as we've heard. We also need some better BBB models in vitro. We are working around this topic as well. We probably also need less severe mouse models, because sometimes the mouse model is really very severe. At birth, the mice has already a lot of neurological damage, so it's really difficult to have an effective treatment in these mice. So of course it's difficult to work with these models. So there are models in which you can induce the disease that have been recently developed. So we're also thinking about using these models in the future that will give us some more time to counteract the disease. So that's it, and this is the work that I'm doing together with Angela Gritti in the San Raffaele Teleton Institute for Gene Therapy in Milan. Dr. Gritti is a co-PI with me on this project. She has a lot of experience with the disease, and I have more experience with the technology, let's say. So we kind of join our efforts, and these are the funding agencies. And of course, this is supported by the ELA and other funding. Thank you.
Thanks a lot, Alessio, for sharing this nice data. And so we will move to our last speaker with, I'm sorry in advance if the pronunciation is not good, Femke Klouwer. We will speak about Zellweger spectrum and how to prepare a clinical trial.
Thank you. I will try to share my screen with you.
It's okay. Yes, we can see.
You see it full screen now?
Yes, perfect.
Okay, thank you. Thank you. Hello, everybody. I would like to thank the ELA organization for the invitation to talk to you today about Zellweger spectrum disorders. And for today, I will tell you something about the clinical overview, but focus on working towards trial readiness and give some news about therapy development. So my name is Femke Klouwer. I'm MD, did my PhD in Zellweger spectrum disorders, and I'm still part of the Center of Excellence for Paroxysomal Disorders within the Amsterdam UMC in the Netherlands. So I will give you a brief biochemical background and a clinical overview, some advice for follow-up, but people who joined earlier, this might already be familiar. So I would like to focus a bit about what is needed for clinical trial readiness for Zellweger spectrum disorders, and give you some therapy development updates. So Zellweger spectrum disorders, they are caused by changes or variants in one of the 13 PEX genes, and most common are variants in PEX1 or PEX6. And since these PEX genes are involved in creating peroxisomes or importing proteins into the peroxisomes, this causes problems. So this is in a normal situation, and here you can see in the Zellweger spectrum disorder where the proteins are not imported into the peroxisome. And because the peroxisomes are not properly formed, this is called biogenesis, basically all peroxisomal functions are affected. So this leads to increased very long chain fatty acids. It leads to accumulation of toxic bile acid intermediates. You get accumulation of branched chain fatty acids, and this is, for instance, the phytanic and the pristanic acid. Also, a plasmalogen deficiency can occur mostly in the bit severely affected patients, and it can also lead to an increased urinary oxalic acid. So how are peroxisomal disorders divided? You basically have the single peroxisomal enzyme deficiency, where only one peroxisomal enzyme is affected, and there are many, but for instance, the XALD. And then you have the peroxisome biogenesis disorders, and these are basically the Zellweger spectrum disorders, and 2 types of RCDP, but I won't talk about that today. So originally, the Zellweger spectrum disorders were basically divided in different subtypes. You had a classic Zellweger syndrome, the neonatal adrenal leukodystrophy, infantile Refsum disorder, and Heimler syndrome that was described in 2015. But yeah, nowadays we basically consider it really as a spectrum of disease. So patients can be anywhere within this spectrum. So it's a bit artificial to really make this subdivision. You can also not clearly distinct them based on the biochemistry or the genetics. And it is good to mention that there are some exceptions. So for instance, PEX 2, 12, 10, 16 can be quite distinct phenotypes. Patients with different kind of symptoms than most Zellweger spectrum disorder patients. So I would not be totally surprised if we would talk about a certain PEX related disease in the future instead of Zellweger spectrum. So then about disease severity, you can basically divide most patients within 3 large group of severity. You have the neonatal infantile presentations. So these are patients that present shortly after birth, for instance, with severe hypotonia. They can have seizures, neuronal migration defects on MRI of the brain. There's usually quite profound failure to thrive, and hepatic dysfunction can occur. Then you have the childhood onset. So the patients that present within childhood, they have usually a bit milder hypotonia. Leukodystrophy, that can be a rapid onset, can occur. There is usually developmental delay, also failure to thrive, but less profound than in the neonatal infantile group. And there's also hepatic dysfunction. Then there is a group of adult onset, and that can also be at a quite later age. They tend to have developmental delay, but there are also cases with normal intellect. There's usually a neuropathy, ataxia, leukodystrophy can occur, but usually more slowly progressive, and also adrenal insufficiency. And independent of severity, most patients usually are suffering from a hearing deficit, retinitis pigmentosa, and also enamel abnormalities. And it is quite important that a treating physician realizes to which category a certain patient belongs, because that is important in order to be fully aware of which problems are likely to occur or very unlikely to occur in this course of disease, since it's so heterogeneous. Dependent on the severity, patients can also look quite different. This is, for instance, a girl that presented with a quite severe phenotype, so that we would probably call a classic Zellweger syndrome in the earlier days. And what is striking is that this child has a very big, large anterior fontanelle, which is really reaching till the forehead. And that is quite typical for Zellweger spectrum disorders. Then there's a girl that is a lot milder affected. She has slight dysmorphic features. And here's a boy that almost didn't have any dysmorphic feature at a younger age, and a bit more profound at a later age. So how is the diagnosis established? Usually with metabolite screening in plasma and urine. So this can be, for instance, very long chain fatty acids, the bile acids, the phytanic acids, etc. Then usually genetic analysis of the PEX gene is performed. But nowadays, more and more often, patients are actually diagnosed by whole exome sequencing, or even as an accidental finding in newborn screening for X-linked adrenal leukodystrophy, where they screen with very long chain fatty acids and then also are able to pick up Zellweger spectrum disorder patients. But of course, in these cases, the diagnosis still needs to be confirmed with, for instance, metabolite screening or genetic analysis. Yeah, then the prognosis is, very depending on the severity, of course. In general, you can say that patients that present with a classic Zellweger syndrome phenotype, are at risk to die within the first year of life. But patients with a milder phenotype can definitely live into adulthood. This also depends a bit on the CACH gene. And there are also rare cases of normal intellect. That is also usually genotype specific. This can, for instance, be patients with a homozygous G843D variant in PAX1, but also some PAX10 patients, for instance. But however, independent of the severity, it is unfortunately, by definition, a progressive disease. So about therapy. At this point, no curative therapy is available, unfortunately, but there are some supportive treatment management that are good to know of. You can supplement, for instance, fat-soluble vitamins. You can treat adrenal insufficiency that can occur, of course, hearing aids, glasses, feeding tube, etc. So I would like to mention at least a few. Low fat soluble vitamins, which are A, D, E, and K, are very common in Zellweger spectrum disorders, also in the milder phenotypes, and this can of course be supplemented. Low vitamin K levels can cause easy bruising, but also severe bleeding in patients with a more severe phenotype, so it's good to be aware of. So we generally advise to do a laboratory checkup of these at least once a year. Then there are the dietary management options. In some patients where there is an elevated phytanic acid, you can start a phytanic acid restricted diet, but in most patients it's not necessary. So it's really good to check these levels before you initiate a phytanic acid restricted diet. For more severely affected patients, of course, a gastrostomy tube can be needed. And at nowadays, DHA supplementation, which is an omega-3 fatty acid, is no longer advised. This was the case in earlier days. Then about adrenal insufficiency. About a third of patients, dependent a bit on which group you're looking at, develop adrenal insufficiency. The adrenal glands normally should produce stress hormones, but if you don't, you can get symptoms like fatigue, but also weight loss, nausea, vomiting, low blood pressure. So this can be quite dangerous. And some patients might be asymptomatic in the beginning. And it's easily treated with hydrocortisone. So also we advise to screen for this adrenal insufficiency, for instance, once a year. Then we know that in recent years, a few patients with Zellweger spectrum disorder develop liver cancer. And it doesn't happen a lot, but it's probably a long-term effect of liver cirrhosis that you are at risk to develop liver cancer. So that is why we also advise, in case of liver cirrhosis, to screen at least every year for the development of possible liver cancer. Then about therapy. There are actually multiple possible approaches to think of to develop a therapy for Zellweger spectrum disorders. You could, of course, correct a genetic defect. That would be ideal. You could try to improve peroxisomal biogenesis, so the formation of peroxisomes. You could try to supplement the deficiencies, for instance, the plasmalogens. And you could try to prevent the accumulation of the toxins, for instance, the very long chain fatty acids. And actually, at this point, there are many different groups that are studying these different approaches. But let's say we would indeed be able to develop a therapy that would be effective. You also need to be ready to actually perform a clinical trial. And this does need quite some preparation. So what would you need? You would at least need some preclinical studies, so in the lab, that really show that a certain therapy is effective, but also safe. You need different models of disease. So for instance, cell models with Zellweger, you need organ models, or for instance, mouse models with Zellweger. You really need to document the natural history of disease to be able to see if there's any effect if you do a clinical trial. And this is actually one of the points that's quite difficult in Zellweger spectrum disorders, because it's so heterogeneous. So it's quite difficult to predict the course of disease in a certain patient. And that is, of course, important if you want to evaluate whether a certain therapy is effective. Then there are different phenotypes in the various involved PACS genes. So you also probably need to focus on one PACS gene in the beginning. And another problem is that biochemical normalization can occur with age. So it's also not really an option to only look at the biochemical profile when you look at the effect of a certain therapy. And furthermore, you really need international collaboration to perform a clinical trial. You need, of course, enough patients to include in a trial, but you also need a collaboration with patient organizations, since, I mean, it's probably difficult to perform all this. So you really need to focus, and you need patient organizations to decide together what should be the primary focus. And that's also because one of the problems, of course, with this is money. It's difficult to get enough money, especially in a rare disease like Zellweger spectrum disorders. So you really need to focus on a certain therapy approach. So what is currently ongoing in this? There are multiple natural history studies ongoing. There is a recently started natural history study that is led by the Global Foundation for Peroxisomal Disorders, the GFPD. And there are two already long-running prospective natural history studies going on, both in the Braverman group in Canada and also Amsterdam. So also quite a lot is already published about the natural history. Then there are multiple preclinical studies ongoing. There's the retinal gene therapy, and I will tell a bit more about that. There are several small compound screenings performed. And we actually in Amsterdam have now a neuronal induced pluripotent stem cells available since 2024 to also use, for instance, for screening purposes or to evaluate a therapy, at least in the preclinical stage. Then there's the NIH-funded R24 research that was done by Joe Hasia for actually a lot of different mouse models for peroxisomal disorders. And this is in place since 2022. So that's already available. And then there was the GFPD hosted FDA-led patient-focused drugs development meeting in June 24. And this was actually meant to, together with a large group of families and patients, establish what should be actually the focus of all these different research groups doing therapy development research. So what would the parents and the patients want to see as a focus? Yeah, so then a bit more about the retinal gene therapy, and this was actually a great work done by Catherine Aguirre together with a group of Nancy Braverman. And the idea behind this is because the retina represents an ideal for target for gene therapy, since it's quite easy accessible. So it's a good start, to say like that. It was also prioritized for treatment by the patient organizations. And that was also because a lot of families and patients expressed that, if you would be able to retain or even recover vision, that could also improve communication, learning, mobility, and autonomy, for instance, for patients with the Zellweger spectrum disorder. Furthermore, all PAX1 patients do develop retinal degeneration, so a lot of patients would potentially benefit from such therapy. And PAX1 gene changes are also the most common cause of Zellweger. So how does this work? You basically have the correct piece of DNA. So this could, for instance, be the PAX1 gene. And this is inserted into a vector, usually a viral vector. And this vector, then containing this correct piece of PAX1, is injected into the subretinal space. And thereby the correct piece of PAX1 is delivered to the retinal cells. So what did Catherine and her group find earlier? They already completed the retinal gene therapy testing in a PAX1 G844D mouse model. And this is actually a mouse model that is similar to the G843D variant in humans. And they found, which is really great, that the retinal response and also the functional vision improved in these mice. So these mice have Zellweger spectrum disorder, and also the retinal structure was preserved. So they did commence a manufacturing process for clinical grade gene therapy vectors, so for humans. And they are currently planning safety studies, and hopefully this is to start in the autumn of 2025. So they need to study where in the body the vector goes after the retinal injection. So this is for safety purposes, but also determine a safe dose. Yeah, and after that, you could start to think, yeah, can we actually prepare for a clinical trial? Well, for that, you do need a good natural history of vision. So this is currently going on. And also you need to establish which patients would benefit and how do we measure success. And of course, another problem with this is that gene therapy development costs a lot of money. So this is another difficult point. So to conclude, although no therapy exists, there are definitely important management options in Zellweger spectrum disorder to be aware of. Working towards clinical trial readiness is important for potential future therapies, and there are quite some research is currently being done in potential future therapies, and international collaboration is of key importance for this. I would like to mention two important patient organizations. So I've already mentioned the GFPD. They are US based, but operate globally. So if you are a family or patient not aware of them, you can definitely look at their website. And also for families who are UK based, there is Zellweger UK.
Thank you.